Hello everyone, thank you for joining my talk. I'm going to present our study with the title of Sexual Selection on Jumping Spider Color Pattern, Investigation with a New Quantitative Approach. At first, I would like to provide some background information regarding animal coloration. Colorful animals are found in wide range of taxa, and color pattern can be a signal in intraspecific interaction, conveying information regarding body condition, nutrition, age, fighting ability, and so on. Besides, sexual dimorphism is also commonly found. So here we raise two questions. First, color pattern could be relatively complex, and how should we measure and quantify them? Second, the color pattern could be sexual selected, and we need evidence from behavior tested, such as female matrice and male contest. For quantifying color pattern, at first we need to keep in mind that the quantification should occur from animal perspective. That is, how animal assess color pattern, but not our human being. Animal may assess information from a particular color pattern component. For example, the cosmophysis Bernetta here is having a stripe on its abdomen, which can be considered as an individual color patch. It contains spectral information of hue, saturation, brightness, and spatial information of shape and size. Animal may also assess information from gross color pattern, which is the combination of color patches. Like the peacock spider here, multiple color patches can be found on its abdomen. Information may convey through within pattern contrast and pattern geometry, and when taking background into consideration, the animal background contrast may also be assessed. Besides, information regarding animal visual system, such as spectral sensitivity and spatial acuity, is also necessary. Luckily, now we are having many recent tools for analyzing animal color pattern, such as QCPA, Pebble, Patternize, PetGeom, and etc. So here I would like to introduce the study object of our work, which is Siler semiglaucus. Siler semiglaucus is a kind of colorful jumping spiders, which lives in South Asia. Here we show the photo of both male-female interaction and male-male competition. Both female and male in this species is colorful, but there is still sexual dimorphism in color pattern. Both female and males frequently tilt their abdomen in intraspecific interactions, so we assume that abdomen color pattern plays a role in sexual interactions. So here comes our hypothesis and predictions. We hypothesize that color pattern evolves through sexual selection in this species, and we have made three main predictions. First, males exhibit more conspicuous color pattern than females. Second, females prefer males bearing greater blue and red color pattern contrast as the within pattern conspicuousness increase. Third, males with more red coverage are more likely to win contests, as red may be perceived as a warning signal or as a warning signal of opponent. We have then quantified both the sexual dimorphism in individual color components and gross color pattern from a statistic perspective. For the individual color patches, we classified the abdomen colors in 5 groups, namely red, blue, black, silver, and iridescent. For the gross color pattern, we used the visual contrast analysis and the boundary strings analysis to quantify the visual contrast and the boundary strings of abdomen color pattern. We used the Mika toolbox for treating the multiple spectral photographs and the QCPA framework for the pattern measurements. We then analyzed the output of individual color patches from QCPA with PCA. We particularly note the two chromatic colors, the red and blue here. According to the PCA results, females and males are having similar red patches as the two sexes cannot be much distinguished from the PC1. As a contrast, male is having smaller but brighter blue patches than females, and the two sexes are clearly distinguished in the PCA result plots on the right. In addition, males are having smaller blue patch but larger red patch comparing to females. We analyzed the QCPA output of gross color pattern with PCA as well. From the visual contrast and analysis result, we noted that there is a stronger sexual differences in global within pattern contrast, which was calculated based on the relative abundance of different color patches. For the boundary strings analysis result, the local within pattern contrast, which was calculated based on the color adjacency of pattern components, is less different between the two sexes. With noting the difference between male and females, especially for their red and blue coverage, we manipulated the male spiders with acrylic paint to 5 different groups with different red coverage, blue coverage, and contrast. The contrast value here is an indicator of relative abundance of red and blue. So when the contrast value is big, it means that the red and blue is equally abundant. And when the contrast value is small, it means that the spider is bearing sole color, which is either completely red or completely blue. We then involve the manipulated male spiders in both female matrices, where female is offered the choices of 2 males, and male-male competition, where 2 males are putting together for their contest, to investigate the role of color pattern in males of this species. So in further data analysis, we only include red coverage and contrast, as blue coverage was strongly negatively correlated with red coverage. We recorded female attention time as the approximate of female interest to males. We construct multiple models with different predictors. We selected the best model where red coverage and contrast jointly predict female interest. So female prefer males with larger red coverage and small contrast the most. Interestingly, color pattern does not affect male contest outcome. So neither red coverage nor contrast predict the outcome of male-male competition in our study. Here we draw conclusions of our study. First, there exists sexual color pattern dimorphism in Siler semiglaucus. The dimorphism is greater in blue color patch than red and greater in global contrast than local contrast. Second, the observed sexual color pattern dimorphism may have evolved through female matrices but not male-male competition. Third, and the most important, we highlighted the importance of the spatial features and relative abundance of colors and patterns in sexual selections. Animals may not only assess a single color pattern component at one time, but may take features of multiple components into consideration simultaneously. That's all of my talk. We'd like to thank Singapore Ministry of Education and National Natural Science Foundation of China for providing funding for this study. We would also like to thank the people listed here for their contribution. Finally, we appreciate the opportunity that Animal Behavior Society provided so that we can share our study in this conference. Thank you.